Welcome to For Science Salah Lotfy channel, where we explore groundbreaking research with real impact on health, economy, and society. Severe bleeding remains one of the most urgent medical challenges worldwide. From road accidents to surgical operations, from natural disasters to battlefield injuries, hemorrhagic shock continues to claim countless lives. In fact, uncontrolled bleeding is responsible for nearly 35% of trauma-related deaths, with many occurring before medical treatment can even begin. To address this urgent problem, Egyptian researchers have developed an innovative solution: a new hydrogel-based sponge dressing. What makes it unique is the combination of advanced polymer chemistry and natural plant extracts, brought together using eco-friendly gamma irradiation technology. The sponge is made from hydroxypropyl methylcellulose, agar-agar, and carbopol, enhanced with extracts from medicinal plants such as Miswak, Yarrow, shepherd's purse, and horsetail. These plants are well known for their natural healing and antibacterial properties. Together they create a dressing that can rapidly absorb blood, activate platelets, and stop bleeding in seconds. Laboratory and animal studies confirmed impressive results: rapid blood and plasma absorption, concentrating clotting factors at the wound site, strong antibacterial action against E. coli and Staphylococcus aureus, reducing infection risks, biocompatibility and safety with no toxicity to normal cells, and most importantly, in a bleeding liver model, blood loss was reduced dramatically, with hemostasis achieved in as little as 8 seconds compared to 50 seconds for conventional hydrogels. But the importance of this research goes beyond the lab. Medically, it offers a life-saving tool for emergency rooms, ambulances, and disaster response teams. Economically, it reduces dependence on imported wound care materials, while opening opportunities for a new Egyptian-made biomedical industry. Socially, it strengthens national healthcare, improves survival rates, and positions Egypt as a leader in radiation technology and biomaterials. This research is a clear example of how science can save lives while boosting economic and social development. For more details on this study and other groundbreaking innovations, stay tuned only on For Science Salah Lotfy channel.
Welcome to For Science Salah Lotfy channel, where science meets curiosity. في هذه القناة لا نكتفي بعرض الأبحاث العلمية، بل نترجمها إلى المعرفة التي تصل إليك أينما كنت. Our mission is to make global scientific research understandable, accessible, and multilingual. جميع الفيديوهات العلمية هنا مترجمة إلى اللغة العربية، بالإضافة إلى لغات عالمية أخرى: French, Spanish, German, Urdu, and more. Because science speaks every language. نعم، العلم يتحدث بكل اللغات، ونحن نحرص أن يصلك بلغتك. If you're passionate about science, you're in the right place. اشترك الآن وابدأ رحلتك مع العلم خطوة بخطوة. For Science Salah Lotfy channel, العلم للجميع.
Welcome to For Science Salah Lotfy channel. In today's episode, we spotlight a remarkable piece of Egyptian scientific research recently published in the International Journal of Biological Macromolecules, Volume 292, 2025, Article number 139132. The study is titled Radiation Development and Hemostatic Performance of Innovative Hydroxypropyl Methylcellulose-Based Sponge Dressings for Controlling Severe Hemorrhagic Wounds. It addresses one of the most urgent challenges in modern medicine: how to stop life-threatening bleeding quickly and safely. This groundbreaking research was carried out by 3 distinguished scientists from the National Center for Radiation Research and Technology at the Egyptian Atomic Energy Authority in Cairo: Professor Dr. Amany Ismail Raafat, Polymer Chemistry Department; Professor Dr. Amr El-Hag Ali, Polymer Chemistry Department; Professor Dr. Asma Abu Bakr Hassan, Radiation Biology Department. Together, they combined expertise in polymer chemistry and radiation biology to deliver a powerful medical innovation. Their work focused on developing a new class of sponge-like hydrogel dressings made from hydroxypropyl methylcellulose, agar-agar, and carbopol. Using gamma radiation, they created a cross-linked, highly porous material capable of rapid fluid absorption. But the innovation doesn't stop there. These sponges were enriched with natural medicinal plant extracts, including Miswak, Yarrow, shepherd's purse, and horsetail, plants long known for their antibacterial and healing properties. The results were striking. The dressings absorbed blood and plasma rapidly, concentrated clotting factors, and triggered platelet adhesion, all key steps in halting bleeding. Laboratory assays showed enhanced protein absorption and stronger clot formation compared to standard hydrogels. Antibacterial tests confirmed activity against dangerous microbes like E. coli and Staphylococcus aureus. In animal models, the difference was dramatic. Bleeding stopped in just a few seconds with minimal blood loss, compared to much longer times using conventional methods. This study demonstrates more than just scientific excellence. Medically, it provides a rapid, life-saving tool for trauma care, surgery, and battlefield medicine. Economically, it reduces the need for costly imported wound dressings and opens the door for Egypt to produce advanced biomedical products locally. Socially, it strengthens healthcare systems, improves survival rates in emergencies, and highlights Egypt's leadership in radiation technology and polymer-based innovations. This pioneering work by Professors Amany Ismail Raafat, Amr El-Hag Ali, and Asma Abu Bakr Hassan showcases how Egyptian science can transform healthcare and save lives. To learn more about this and other groundbreaking studies, stay tuned here on For Science Salah Lotfy channel. Welcome to For Science Salah Lotfy channel. Today we explore a cutting-edge scientific development designed to tackle one of the most urgent challenges in modern medicine, controlling severe hemorrhagic wounds. Globally, traumatic injuries and severe hemorrhagic wounds caused by natural disasters, wars, traffic accidents, or complications in operating rooms, especially during childbirth, remain some of the toughest humanitarian and economic problems. The World Health Organization reported that injuries accounted for 9% of global deaths in the year 2000. Every year, trauma claims over 5 million lives worldwide. By 2030, it is expected that traffic accidents, violence, and suicides will make up a significant portion of trauma-related deaths, further burdening healthcare systems and societies. Alarmingly, hemorrhage contributes to nearly 35% of these deaths, with up to half occurring before effective medical treatment can even begin. To address this deadly challenge, researchers turned to an eco-friendly scientific tool, gamma-ionizing radiation. Using this technology, they developed innovative hydrogel-based sponges designed specifically to stop severe bleeding and accelerate wound healing. These dressings are not ordinary. They are created from natural polymeric networks such as hydroxypropylmethylcellulose, agar-agar, and carbapol, which together form highly absorbent, porous structures capable of holding large amounts of fluids. Their three-dimensional design closely resembles the body's natural extracellular matrix, making them ideal for supporting cell growth, clot formation, and tissue repair. But the real innovation lies in combining this hydrogel technology with traditional medicinal plant extracts. Extracts from Miswak, Yarrow, shepherd's purse, and horsetail were incorporated into the sponge matrix. These plants are rich in natural compounds such as tannins, flavonoids, alkaloids, and essential oils, well known for their antibacterial, antioxidant, and healing properties. They not only help control bleeding, but also prevent infections, reduce inflammation, and speed up tissue regeneration. This dual action, hemostasis and antimicrobial protection, makes the dressings highly effective for trauma management. To test their effectiveness, the researchers conducted a series of in vitro and in vivo evaluations. The sponges showed high swelling capacity, absorbing fluids rapidly, and concentrating clotting factors at the wound site. Cytocompatibility tests confirmed that the materials were safe and non-toxic to living cells. Antibacterial studies demonstrated strong activity against both gram-positive and gram-negative bacteria. Finally, in animal experiments using a rat liver hemorrhage model, the results were remarkable. Bleeding stopped in seconds with significantly reduced blood loss. This proves the strong hemostatic potential of the new hydrogel sponges. This innovation is more than just a scientific achievement. Medically, it provides an advanced solution to save lives in emergency rooms, ambulances, and even on battlefields. Economically, it reduces dependence on imported wound care products and opens opportunities for a new Egyptian-made biomedical industry. Socially, it improves healthcare outcomes, lowers mortality rates, and highlights Egypt's growing leadership in the field of radiation technology and biomaterials. By blending modern science with traditional medicinal knowledge, this research paves the way for safer, more effective, and affordable medical treatments. This breakthrough research shows how science can address some of humanity's greatest challenges. Stay with us on For Science Salah Lotfy channel as we continue to explore innovations that save lives and build a better future. Welcome back to For Science Salah Lotfy channel. In our previous episode, we introduced you to an innovative study that developed advanced hydrogel-based sponge dressings for controlling severe hemorrhagic wounds. Today we go deeper, exploring the scientific methods, materials, and experimental procedures that made this research possible. To build these advanced dressings, the research team used a combination of synthetic polymers, natural agents, and medicinal plants. Hydroxypropyl methylcellulose, or HPMC, was sourced locally from El Nasser Pharmaceutical Chemical Company in Egypt. Agar-agar, commonly used in microbiology, came from Fluka, and carbapol 940 was obtained from Sigma Aldrich in Germany. But the heart of this innovation lies in natural medicinal plants. Dried and crushed leaves of Miswak, Yarrow, shepherd's purse, and horsetail were purchased from local Egyptian herb markets. These plants were carefully chosen for their proven hemostatic and antibacterial properties. Together, these materials formed the foundation for the hydrogel sponge dressings. For in vivo testing, Swiss albino rats weighing about 120 grams were obtained from the National Center for Radiation Research and Technology, or NCRT, in Cairo. The animals were kept under standard laboratory conditions with controlled diets and environments, ensuring reliable and ethical testing conditions. This acclimatization period was critical to prepare them for the experimental procedures. The hydrogel samples were then subjected to gamma irradiation using a cobalt-60 source at NCRT. With a dose rate of 1.67 kilograms per hour and a total dose of 15 kilograms, this step achieved two goals simultaneously: cross-linking the polymer network to form stable hydrogels and sterilizing the material in one eco-friendly process. Medicinal plant extracts were prepared using hydroalcoholic extraction. The crushed leaves were soaked in 60% ethanol for 72 hours, filtered, and then concentrated under vacuum using a rotary evaporator. The resulting dry extracts were mixed into HPMC solutions containing different concentrations of agar-agar and carbapol. The mixtures were then packed, irradiated, and finally freeze-dried for 12 hours to produce porous, sponge-like hydrogel dressings. To understand their performance, the hydrogels underwent several evaluations. Gel fraction tests measured cross-linking efficiency. Swelling capacity tests examined how well the sponges absorbed fluids like saline, blood, plasma, and simulated body fluids. Porosity and density measurements were conducted using ethanol displacement. Hydrolytic degradation tests monitored stability over time in phosphate-buffered saline. These tests confirmed that the hydrogels had high absorption, controlled porosity, and sufficient stability for medical applications. Scanning electron microscopy revealed the internal structure of the sponges, showing highly porous surfaces ideal for platelet adhesion and clot formation. Platelet-rich plasma experiments confirmed that platelets strongly adhered to and aggregated on the surface, a vital step for rapid hemostasis. Ultraviolet spectrophotometry and protein absorption tests using bovine serum albumin further demonstrated the hydrogel's ability to bind essential proteins, enhancing the clotting process. Next, the blood clotting index was measured by applying blood samples and calcium chloride to the hydrogels. The results showed fast clot initiation and stabilization. Plasma coagulation assays including prothrombin time, PT, and activated partial thromboplastin time, APTT, confirmed that the sponges significantly improved coagulation efficiency compared to controls. This was critical in demonstrating that the materials could work even in challenging medical conditions. Safety was equally important. Using an MTT cytotoxicity assay with fibroblast cell lines, the researchers proved that the sponges were non-toxic and compatible with living tissues. Antibacterial activity was then tested against Staphylococcus aureus and Escherichia coli, two of the most common pathogens in wound infections. The results showed strong bactericidal potential, confirming that the dressings could reduce the risk of infection while stopping bleeding. Perhaps the most critical test came in the in vivo experiments. Using a rat liver hemorrhage model, researchers applied the sponges directly to bleeding wounds. Blood loss was carefully measured, and the time to achieve hemostasis was recorded. The treated groups showed dramatically reduced bleeding times compared to untreated controls, with bleeding stopped in seconds rather than minutes. These results validated the sponge's real-world hemostatic potential. To confirm the biochemical effects, calcium and transferrin levels were measured, showing positive impacts on the hemostatic process. Histopathological examinations of liver tissues revealed normal healing patterns with minimal inflammation, further demonstrating the safety and therapeutic potential of these hydrogel dressings. All experiments were conducted under strict ethical guidelines, with approval from the Research Ethics Committee at NCRRT. Blood samples from healthy volunteers were collected with informed consent, and all animal studies followed international standards for humane research. This meticulous scientific journey, from material selection to radiation processing, lab testing, animal models, and biochemical analysis, demonstrates the depth and precision of Egyptian research in biomedical innovation. These hydrogel sponges represent a powerful, eco-friendly, and life-saving tool for trauma care and emergency medicine. Stay tuned on for Science Salah Lotfy channel as we continue to bring you closer to the science shaping our future. Welcome back to For Science Salah Lotfy channel. We explored how innovative hydrogel dressings were developed using gamma irradiation and natural medicinal plant extracts. Today, we dive into the heart of the study, the results and discussion. This is where the science proves its real impact. When natural polysaccharides such as hydroxypropyl methylcellulose, agar-agar, and carbapol are exposed to gamma irradiation, two key processes can occur: chain scission or cross-linking. In this case, irradiation caused the formation of highly reactive hydroxyl radicals. These radicals attacked the polymer chains, creating free macroradicals that recombined into a strong, three-dimensional network. The outcome was a stable hydrogel structure with enhanced gelation and durability. The gelation degree reflects how well cross-linked and insoluble the hydrogel becomes. The study showed that increasing carbapol content improved the gelation degree, forming denser and more reliable networks. On the other hand, higher agar-agar content reduced gelation due to radiation-induced degradation. This balance between components determined the strength and stability of the final sponge dressings. When medicinal plant extracts, Miswak, Yarrow, shepherd's purse, and horsetail were added, the gelation degree slightly decreased. Why? Because compounds like flavonoids and phenols interfere with the radiation cross-linking process. Some water-soluble extracts even leached out during testing. Still, the decrease was minor, and the plant extracts introduced valuable bioactive properties, including antibacterial and hemostatic effects. Porosity plays a vital role in hemostatic dressings. The hydrogels displayed porosity levels above 90%, with very low densities. This means a huge surface area is exposed to fluids, allowing faster blood absorption and clot formation. The porous structure also forms a sticky gel layer upon contact with blood, sealing wounds and accelerating hemostasis. The sponges were tested in different fluids: distilled water, saline, and simulated body fluid. In all cases, they swelled rapidly, absorbing large amounts of fluid. After just 1 hour, swelling degrees reached up to 19 grams of fluid per gram of material. The Miswak-based dressing showed particularly high swelling ability. This rapid uptake mimics real wound conditions, where the dressing must immediately absorb plasma and concentrate clotting factors. Another critical property is biodegradability. Ideally, a hemostatic dressing should degrade naturally, avoiding the need for surgical removal. Over 7 days in phosphate-buffered saline, the base hydrogel degraded almost completely, while the plant-enriched versions degraded slightly more slowly. The natural extracts reinforced the network, providing stability but still maintaining safe biodegradability. The ultimate test was blood absorption. Within just 2 minutes, the dressings absorbed up to 4 grams of blood per gram of material. After 2 hours, absorption increased dramatically, reaching as high as 15 grams per gram. Plasma absorption was equally impressive, confirming that the dressings not only captured blood but also concentrated essential clotting components. This rapid action is critical for stopping severe hemorrhage. In vitro and in vivo tests further validated their performance. Platelets adhered strongly to the sponge surface, triggering clot formation. Antibacterial assays showed effective action against both Staphylococcus aureus and Escherichia coli. These dual functions, stopping bleeding and preventing infection, make the hydrogel sponges powerful tools for trauma care. In animal trials using a rat liver hemorrhage model, the sponges proved life-saving. When applied to a bleeding wound, they reduced both blood loss and clotting time compared to untreated controls. Bleeding was stopped in seconds. Biochemical tests confirmed improvements in calcium and transferrin levels, key indicators of hemostasis. Histological studies of liver tissues showed normal healing, minimal inflammation, and no toxic effects. These findings highlight the promise of radiation-developed hydrogel dressings enriched with natural plant extracts. They combine high porosity, rapid absorption, biodegradability, antibacterial action, and strong hemostatic performance. Medically, they could save countless lives in accidents, surgeries, childbirth, and battlefields. Economically, they reduce dependence on imports and open the door for Egypt to lead in biomedical manufacturing. Socially, they strengthen national healthcare and showcase the global relevance of Egyptian science. This research proves that when modern radiation technology meets the healing wisdom of nature, the result is innovation with the power to save lives. Stay tuned to For Science Salah Lotfy channel for more insights into science that matters for Egypt and the world.
Welcome back to the For Science Salah Lotfy channel. In this episode, we continue our exploration of advanced hydrogel-based hemostatic dressings, an innovation designed to save lives during severe bleeding events. Previously, we discussed how hydrogels were synthesized, their porosity, and their ability to absorb fluids. Today, we will go deeper into their plasma absorption capacity, thrombogenic performance, protein interactions, platelet adhesion, and in vivo experiments. The plasma absorption ability of these hydrogel dressings is remarkable. Compared to the parent hydrogel, which absorbed about 13 grams of plasma per gram of material, the modified dressings absorbed up to 22 grams per gram, depending on the type of medicinal plant extract added. Why does this matter? Rapid plasma absorption is crucial. It prevents uncontrolled bleeding and supports platelet adhesion, thrombin generation, and clot formation. Another critical parameter is the blood clotting index, or BCI. The parent hydrogel showed weaker hemostatic activity with a higher BCI value around 13. But when plant extracts such as Miswak, Yarrow, shepherd's purse, or horsetail were incorporated, the BCI dropped significantly to values between 7 and 9. A lower BCI means stronger clot formation. This confirms that the modified dressings accelerate thrombosis more efficiently than the base hydrogel. Scanning electron microscope images give us a closer look. On the parent hydrogel, only a few red blood cells were observed. But on the modified dressings, blood cells aggregated tightly, forming strong clots. Interestingly, many red blood cells remained intact with their characteristic donut shape, while others showed surface crimping due to dehydration, evidence of rapid clot stabilization. This structural reinforcement mimics the natural fibrin network, making the clot both stable and durable. Protein adsorption is another factor. Using BSA as a model protein, the modified dressings showed a significant increase in protein absorption, particularly with Miswak extract reaching nearly 400 milligrams per gram. When blood proteins quickly bind to the surface of a dressing, they activate platelets and accelerate the coagulation cascade. This leads to the rapid formation of a protective barrier at the wound site. The study also evaluated clotting time using prothrombin time, PT, and activated partial thromboplastin time, APDT. Compared to the control group, which recorded 36 seconds, the modified dressings dramatically shortened clotting time. For example, Miswak incorporated hydrogels reduced APTT to just 5 seconds and PT to only 3 seconds. These results highlight the powerful role of plant-derived bioactive compounds in accelerating the clotting process. Platelet adhesion is key to primary hemostasis. The parent hydrogel showed only 5% platelet adhesion, but the modified versions demonstrated far higher values. Miswak at 22%, Horsetail at 19%, shepherd's purse at 14%, and Yarrow at 12%. SEM images confirm heavy platelet activation, showing pseudopodia formation spreading across the hydrogel network. This spider-like extension of activated platelets signals robust clot initiation. Safety is a priority in medical applications. Using an MTT assay on normal fibroblast cell lines, the developed dressings showed high cell viability, ranging from 90% to 66%, depending on extract concentration. These results confirm that the materials are biocompatible and safe for wound care. Another advantage of these dressings is their antibacterial activity. Against both gram-positive Staphylococcus aureus and gram-negative Escherichia coli, the modified hydrogels significantly reduced bacterial colonies, unlike the parent hydrogel, which showed no antibacterial effect. The strongest antibacterial effect was observed in Miswak-based dressings, confirming traditional knowledge of its antiseptic properties. Finally, the research team conducted in vivo tests on rat liver injury models. Compared with untreated control animals, those treated with the developed dressings showed a dramatic reduction in both hemostatic time and total blood loss. Photographs of treated livers clearly demonstrated less bleeding and faster wound closure, proving the effectiveness of these novel hydrogel-based dressings in real biological systems. In summary, these spongy hydrogel dressings, enhanced with natural plant extracts, represent a new frontier in hemostatic technology. They combine rapid blood absorption, efficient clotting, antibacterial properties, and biocompatibility, making them powerful tools for trauma care and emergency medicine, where we will continue uncovering the science behind innovative biomedical materials. You are watching For Science Salah Lotfy channel. Welcome back to the For Science Salah Lotfy channel. Today we conclude our journey into the fascinating world of hydrogel-based hemostatic dressings enhanced with natural plant extracts. In this part, we focus on the biochemical findings, histopathological investigations, and the overall conclusion of the study. When tested in a rat liver injury model, the results were striking. The untreated control group lost about 75 milligrams of blood in 95 seconds, while the parent hydrogel reduced bleeding to 54 milligrams in 50 seconds. But the real breakthrough came with the plant extract dressings. The Miswak-based hydrogel stopped bleeding in just 8 seconds, with blood loss as little as 2.1 milligrams. Horsetail followed closely at 9 seconds, Shepherd's Purse at 10 seconds, and Yarrow at 13 seconds. This dramatic reduction confirms the synergy between the hydrogel's hydrophilic network and the natural hemostatic power of these medicinal plants. Hemostasis isn't just about stopping blood flow, it's a finely tuned biochemical process. Two key markers were measured: calcium and transferrin. Calcium is essential for platelet activation and clot formation. Groups treated with plant-based dressings showed significantly higher calcium levels compared to the parent, hydrogel, and controls. Miswak again showed the strongest effect. Transferrin, a plasma protein critical for both immunity and coagulation balance, was also significantly elevated in rats treated with the plant-enhanced dressings. This means the dressings not only stop bleeding faster but also support the body's natural healing and defense systems. Looking under the microscope, the difference was clear. The untreated livers showed necrosis, hemorrhage, and tissue edema, but in animals treated with Miswak, Yarrow, Horsetail, or Shepherd's Purse dressings, there was evidence of fibrin deposition, collagen formation, and reduced tissue damage. In other words, healing had already begun. So what have we learned? By combining hydroxypropyl methylcellulose, agar-agar, and carbapol with extracts from Miswak, Yarrow, Shepherd's Purse, and Horsetail, scientists developed eco-friendly gamma-irradiated hydrogels that absorb fluids rapidly, shorten bleeding time dramatically, enhance calcium and protein balance in blood, promote platelet adhesion and activation, provide antibacterial protection, and accelerate overall wound healing. These findings point to a promising new generation of topical hemostatic agents, potentially life-saving in trauma care, surgery, and emergency medicine. You've been watching the For Science Salah Lotfy channel, where we explore the science shaping our future in health and technology. Stay tuned for more insights into groundbreaking research. As we conclude today's episode, let's reflect on the significance of this research. The development of hydrogel-based hemostatic dressings, enhanced with natural medicinal plant extracts, represents a powerful union of modern radiation chemistry and traditional healing knowledge. By employing eco-friendly gamma irradiation, these dressings achieve superior blood absorption, rapid clotting, antibacterial activity, and remarkable healing potential. From Miswak and Horsetail to Yarrow and Shepherd's Purse, nature's pharmacy has been harnessed to create materials that could one day save countless lives in surgery rooms, ambulances, and emergency care units. This achievement would not have been possible without the dedication and vision of the research team. Professor Dr. Amany Ismail Raafat, Polymer Chemistry Department, National Center for Radiation Research and Technology, Egyptian Atomic Energy Authority. Professor Dr. Amr El-Hag Ali, Polymer Chemistry Department, National Center for Radiation Research and Technology, Egyptian Atomic Energy Authority. Professor Dr. Asma Abubakar Hassan, Radiation Biology Department, National Center for Radiation Research and Technology, Egyptian Atomic Energy Authority. On behalf of myself, Professor Dr. Salah Lotfy, Professor at the Egyptian Atomic Energy Authority, and on behalf of For Science Salah Lotfy channel, I extend my deepest thanks and appreciation to these outstanding researchers for their remarkable efforts. To our viewers, we invite you to share your thoughts about this research in the comments section below. Your insights and discussions add value to the scientific journey we are building together. Thank you for watching, and stay with us on For Science Salah Lotfy channel for more explorations into science, innovation, and discovery.